Students, this is the NEET preparation classes of biology. The next unit that we are going to see now is cell, the unit of life. So first of all, what is a cell? Cell, it can be very technically defined as the structural and functional unit of a life. Means it is a structural unit because it is the structure in which all functions of life is going to happen. So it is defined as the structural and functional unit of life. This cell was first discovered by a person called Robert Hooke. He discovered the cork cells of plants. So it was a dead cell that he observed first. The first living cell was first observed by a scientist called Antony von Leeuwenhoek, and don't have confusions in his spelling because few books give it as Anton von Leeuwenhoek. Both Antony von Leeuwenhoek and Anton von Leeuwenhoek, it is going to define the same person, and sometimes it can be given as Antony or sometimes as Anton von Leeuwenhoek. And about the cell, how it is functioning and how it is present in a cell, the cell theory was proposed. Cell theory was proposed by two persons called as Schleiden and Schwann, and now currently what is in acceptance, it is called as the modern cell theory. It is actually the theory is given by Schleiden, Schwann and one more person called as Rudolf Virchow. So what does these three persons state about a cell? They have given us three principles. The first one is all living organisms are made of cells. So any living organism, if it is said to be living, it should be made of cells. So all living organisms are made of cells, and all functions of a living organism, are the outcome of the functions of a cell. So when we call a function of a living organism, it means some cells are going to function inside that living organisms, giving the function to that organism. And these two principles was given by Schleiden and Schwann. And the third part of cell theory, which was given by Rudolf Virchow, states, this is a famous statement called as omnis cellula e cellula, which means, this was a statement given by Rudolf Virchow, and this means all cells arise from pre-existing cells. This is what the sentence omnis cellula e cellula means. So this was given by Rudolf Virchow, and these two statements were given by Schleiden and Schwann. Together make up the modern cell theory. When we study about a cell, we have to study about three important characteristics of a cell. The three important characteristics that has to be studied for a cell is its size, shape and its complexity. So three factors or three characters that is very important about a cell. Coming to the size of the cell, the size of the cell is going to vary between one cell and the other cell. No two cells are going to have the same size unless and until they belong to the same species and same group of tissues. And the size of the cell is decided by three factors. Three factors that decide the size of the cell. The first factor, it is called as the Nucleo cytoplasmic ratio. So based on the size of the nucleus and the amount of cytoplasm, the size of the cell would be divided. If the cell nucleocytoplasmic ratio is going to differ, the cell can take up cell division. Similarly, one more factor is surface area to volume ratio. So again, these factors are all going to decide whether the cell has to remain in the same size or they have to change their size. When they go for changing their size, the main thing that they take up to change their size is going to be cell division. And the third factor is going to be the rate of metabolism. So as the metabolic activities increase, if the cell is not able to cope up with the speed of metabolic activities, the cell can divide, increase the number of cells to face those metabolic activities. So this is the three factors that decides the size of the cell. And we can also study some factors about the smallest cell. Smallest cell, when it is written, be clear if it is given without cell wall or with cell wall. If it is a smallest cell without cell wall, it is going to be the mycoplasma, also called as PPLO. This is pneumonia like organisms. We will study the details of mycoplasma in the unit of biological classification. And the smallest cell with cell wall is going to be a bacterial cell. And the largest cell ever known is the egg cell. Egg cell, don't consider the egg, it is the egg cell, which represents the female gamete of ostrich. This is the largest cell ever known. The next factor into consideration is the shape of the cell. Shape of the cell is mainly decided by its outer cover. Since plant cells have their cell wall and animal cells do not have their cell wall, plant cells have more or less common shapes, whereas animal cells, they can take up different shapes. The different shapes, they can be regular in shape, they can be spindle shaped, or they can be rectangular, branched, biconcave shapes, etc. When we see plant cell, they are mostly going to be polygonal or hexagonal in shape, and a different type of shape seen in plant cells are going to be bean shaped cells and dumbbell shaped cells. So these are going to be the different types of shapes that are taken up by the cells. We will study about the examples of all these cells, their functions, where they are present, etc. All those, that is in case of animal cells, we will be studying it in human physiology and structural organization in animals. And in case of plant cells, we will be studying all these shapes when we study about the anatomy of flowering plants. And the next factor that can be considered is the complexity of the cell. Complexity of the cell, it is usually deciding whether the cell is going to be a simple cell or a complex cell. And based on this, there are two types of cells which are called as, one is called as prokaryotic cell, and the second type of cell is eukaryotic cells. Here I have given you some basic differences between a prokaryotic cell and a eukaryotic cell. To start with, what does a prokaryotic cell mean? What does the term prokaryote mean? When we say prokaryote, pro means primitive and karyon means nucleus, means a prokaryotic cell does not have a defined nucleus, whereas eukaryotic cells, they have a well defined membrane bound nucleus. And all these structures, all these characteristics we have written here, we would be seeing it in detail in the same unit. And prokaryotic cells, they do not have membrane bound organelles, whereas eukaryotic cells have them. Ribosomes are going to be 70S type, and here ribosomes are going to be 80S type. And if you don't understand all these terms, what is this 70S, what is this membrane bound, why it is called as well defined, and whether it is not defined, all these we'll be seeing it in detail when we study about all these organelles separately. And they have, prokaryotic cells, they have a single haploid chromosomes, whereas eukaryotic cells, they are going to have multiple chromosomes and they can be diploid or a haploid. And their cell wall is going to be a very complex structure in case of prokaryotes, whereas eukaryotic cells, they have simple cell wall with depositions of substances. And the deposition of substances is mainly with polysaccharides. And prokaryotic cells do not have any cytoskeletal structure. Cytoskeletal structures are structures that give strength to the cell, and prokaryotic cells do not have them, whereas eukaryotic cells have them. And plasmids, they are present in prokaryotes. Most of the prokaryotic cells possess plasmids, and these plasmids are absent in eukaryotic cell, and if at all present, they are going to be very rare. Plasmids are extra chromosomal DNA. We will see it in later classes. Average cell size of a prokaryotic cell ranges between 0.5 to 100 micrometers, whereas in a eukaryotic cell, it is between 10 to 150 micrometers. You can see the basic size of a prokaryotic and a eukaryotic cell, they vary greatly. And prokaryotic cell, they do not have any membrane bound receptors. This can also be, these plasma membrane receptors, they are usually called as the membrane bound receptors. These membrane bound receptors are absent in case of prokaryotic cell, whereas these receptors are present in prokaryotic cell, and the receptors are usually sterols. In case of, this is an example of a membrane bound receptor made of sterols. Here we'll start with the characteristics of prokaryotic cell in detail. So prokaryotic cell can be also defined as the bacterial cell. It is a typical bacterial cell which is going to have all the characteristic of a prokaryotic cell. So we'll first see the structures that are present, which are visible one by one, and their characteristics and their functions also. The first outermost cover, which is called as the cell wall. The cell wall of prokaryotic cell is a complex cell wall made of modified sugars, and the cell wall is also called as peptidoglycan layer. Peptidoglycan is made of modified sugars like N-acetyl glucosamine, which is commonly called as NAG, and one more substance called as N-acetyl muramic acid, which is called as NAM unit. This NAM and NAG units in a bacterial cell wall is going to be present alternatively like this, and they usually form a double layer. There can be many layers of these double layers, but when we call a single peptidoglycan unit, a single peptidoglycan unit is going to have two layers of alternatively arranged NAM and NAG. And this NAM and NAG units, these two layers, they are interlinked with the help of amino acids. So these two structures, they are interlinked with amino acids, or we can call it as a peptide chain. And this is the cell wall of a bacteria or a prokaryotic cell. And there is another layer which can be or cannot be present in a bacterial cell. This is called as Glycocalyx. These Glycocalyx, it is found external to cell wall, and this Glycocalyx is of two types. One is called as the slime layer. The other type of Glycocalyx is called as capsule. When it is a slime layer, it is a loosely attached layer, and capsule is a tightly bound layer. And whichever bacteria has a capsule means they are mostly pathogenic. So capsulated bacteria are mostly pathogenic, not always, mostly they are going to be pathogenic. And this Glycocalyx layer, generally what is their function is that Glycocalyx prevents bacterial cell from drying. So when they are exposed to higher temperatures or in unfavorable conditions, they prevent the cell from drying. The next layer is going to be plasma membrane. Plasma membrane, it is a lipid bilayer with proteins and carbohydrates, and they exhibit a fluid mosaic model. We will study in detail about fluid mosaic model when we study about the eukaryotic organelles. And the next part of the bacterial cell is a mesosome. Mesosomes are invaginations of plasma membrane. So why is this invaginations seen as? Mesosomes have all enzymes and the electron transport chain for the respiration process. I hope you know what are the enzymes that are required for respiration and the electron transport chain. In the before unit we have seen it. And mesosomes have one more function. During cell division, that is when the bacterial cell takes up binary fission, during cell division, the DNA, that is the genetic material of the bacterial cell, is usually attached to mesosomes. This prevents the loss of this DNA because it is an abruptly dividing or approximately dividing cell. So mesosomes, the DNA gets attached to this mesosome so that they are not lost. And the next part of the bacterial cell is the cytoplasm. Cytoplasm, they have no membrane bound organelles. We have seen this already. They do not have any membrane bound organelles, and they have what else is present in the cytoplasm is going to be enzymes for metabolic activities, their nutrients, the waste or the excretory substances and gases that are taken in and given out by the cells. These are substances that are going to be present in the cytoplasm of the bacterial cell. The next part is the only structure that is seen in the cytoplasm of bacterial cell is the ribosomes. Ribosomes are going to be 70S type and they are going to be floating in the cytoplasm. We will see the details of these ribosomes later on. The next part that is present in the cell is going to be The genetic material. The genetic material of a bacterial cell is going to be a double-stranded, circular DNA molecule, and the place where this genetic material is present is usually called as nucleoid. The next part is the plasmid. Plasmids, they can be defined as extra chromosomal, circular, double-stranded DNA molecules, and these plasmids, they are going to offer the bacterial cells with some special characteristics. We'll see some examples alone, examples of few characteristics given by plasmids. The first example, the plasmids are called as R plasmids if they are going to give resistance to antibiotics. And the plasmids are called as F plasmids. F plasmids, they are also called as fertility plasmids. The presence of fertility plasmids makes the bacteria suitable for conjugation process. Conjugation is a type of genetic exchange which can be also considered as the sexual reproduction in bacterial cells, and the F plasmids gives that characteristics to the bacteria in which they are present. And one more type of plasmid is also called as Col plasmids. These plasmids, they are going to give resistance to toxins called as coalescens. So coalescens are type of toxins, and these coalescens are actually produced by bacteria like E. coli. This is one example of a bacteria, E. coli. It is not that coalescens are produced by only E. coli. They are produced by a group of bacteria. One example is E. coli. And if Col plasmids are present, those bacteria can be resistant to these coalescens. So these are the basic characteristics of the bacterial cell or a prokaryotic cell. And the prokaryotic cells or bacterial cells, they are classified into two types. This types classification is based on their staining process. Staining means giving color to the cell. So the staining that is used to differentiate between the bacterial cell, it is called as Gram's staining, developed by a person called Christian Gram. This person, he developed a special stain called as Gram's iodine. It is not simply iodine. He named it as iodine, but it is actually combination of iodine with some other substances also. It was called as Gram's iodine. The two types of bacteria they are called as Gram positive and Gram negative bacteria. Gram positive bacteria are those bacteria that can retain Gram's iodine after washing with alcohol. So when a Gram positive, when a bacteria is first stained with Gram's iodine and then washed with alcohol, if the bacteria, even after washing with alcohol, if they are able to retain the Gram's iodine, they are called as Gram positive bacteria. The reason why they are going to retain the bacteria is they have a thick cell wall. The thickness is a cell wall is defined by the number of peptidoglycan layers. These bacteria are known to possess, that is the Gram positive bacteria, approximately around 40 peptidoglycan layers. So because of this many layers, they are able to retain the Gram's iodine. Whereas Gram negative bacteria, they lose Gram's iodine and they take up the counter stain. That is, Gram's iodine usually looks blue in color, and what Christian Gram did was he found some bacteria, they lost this Gram's iodine. So what he did, he stained the bacteria again with another stain, a counter stain, which is called as safranin. And this safranin usually gives pink color to the cell because it is a pink color dye and it gives pink color to the cell. Because Gram's iodine is not lost in Gram positive bacteria, they do not take up the safranin. Whereas Gram negative bacteria, because they lose their Gram's iodine stain, they take up the counter stain and they look pink in color. And why do they lose this Gram's iodine is because their cell wall, usually they have only 1 to 4 peptidoglycan layers. So because of those small number of layers, they easily lose Gram's iodine when it is washed with alcohol. Whereas Gram positive, because they have nearly 40 layers, they do not lose the Gram's iodine. So this is how we differentiate between Gram positive and Gram negative bacteria. And the next part of the cell is going to be structures that are attached to the external surface of the cell, which can be flagella, pili, or fimbriae. Here flagella are going to be long hair-like outgrowths, whereas pili and fimbriae are short hair-like outgrowths. Flagella helps in, so here I am writing it here for flagella. Flagella is going to help in locomotion, that is the movement of the organism from one place to the other. Pili helps in the conjugation process. They are formed usually only, pili formation happens only in bacteria that contain this F plasmids. So it helps in conjugation process. And fimbriae, they help in attachment to surfaces. So these are structures that are present in the externally to their cell wall. They are usually attached from the plasma membrane and they extend out. And the flagella numbers are usually countable, that is their numbers are less and they are be countable. Pili also similarly can be countable because they are formed only during conjugation. Whereas fimbriae, they are going to be very large in number and they are very numerous and it is uncountable, and they are going to completely cover the external surface of the bacterial cell. This diagram shows you a typical bacterial cell, and we will just mark the parts so that we are clear with what we have already seen. So the external cover, you call it as the cell wall, which is a peptidoglycan layer, and the next part is going to be the plasma membrane. So plasma membrane has some invaginations, and these invaginations are called as the mesosomes. So these are mesosomes, and this part, what is present found internally, is the cytoplasm, and the only structure that can be seen in the cytoplasmic part are the ribosomes. So it has a double-stranded circular DNA molecule, which is the genetic material present in a region called as nucleoid. And the cells also have the extra chromosomal DNA, which is called as the plasmids. And these long hair-like structures, what we have drawn here, represents the flagella. And I have not shown you pili here, and pili, if at all if it is formed, they are going to be smaller structures that are going to be formed like this, and these pili are usually extensions of plasma membrane. They are going to grow out like this to form these extensions, which can be called as the pili, which help in the conjugation process. And these small hair-like outgrowth, I have shown it only one part of the cell. Actually, it covers a complete layer of the cell. And these structures, you call it as fimbriae, which helps the cells to attach to the surfaces where they are present. So these are the parts of a bacterial cell or a typical prokaryotic cell. So we have completed with the prokaryotic cell structure, and coming to the eukaryotic cell, we will first see some basic differences between a plant cell and animal cell, because a typical eukaryotic cell stands with the plant and animal cells. So we will see some basic differences between the plant and animal cells. Plant cells, they are usually larger than animal cells, whereas animal cells are going to be smaller compared to the plant cells. It is not that they are always smaller, comparatively they are going to be smaller. And the shape of plant cells is most probably hexagonal or polygonal, and in case of animal cells they are going to have different shapes. The reason for this hexagonal or polygonal fixed shapes is because of their cell wall, whereas animal cells, the cell wall is absent, so they can take up any shape they need. Plant cells have plastids, and the plastids are absent in the animal cells. Plant cells have a single large and a permanent vacuole with an extra special covering for that vacuole called as tonoplast, whereas animal cells, they do not have a tonoplast, that is their vacuoles are going to be only smaller and temporary vacuoles, and they do not have that tonoplast also. But the difference is the plant cell is going to have a single vacuole, whereas the animal cells has numerous vacuoles in them. And plant cells do not have centrosomes and centrioles, and animal cells have centrosomes with 2 centrioles in them. They have a single centrosome with 2 centrioles inside them, and we will see the details of all these structures in the later classes. And the nucleus of the plant cell is usually found at the periphery, because this vacuole pushes the nucleus to the periphery of the cell, whereas animal cells, they have their nucleus found at the center. Plant cells rarely have lysosomes, whereas animal cells, most of the animal cells have lysosomes in them. And plant cells store starch granules, and animal cells stores glycogen granules. So these are the basic differences between the eukaryotic cells, that is typical eukaryotic cells of plant cells and animal cells, the basic differences between them. Now we will see about the structure of a plant cell separately and structure of the animal cell separately. Here we will just see the diagrammatic representation of a plant cell and an animal cell. This is because when we study, we will just mark the parts of the plant cell, what are present in plant cell, important organelles and structures formed. Later on we will study each and every structure in detail with their functions, characteristics, as well as how they are seen, what number is present, all these. So the outermost cover is going to be the cell wall in case of a plant cell. The next part is going to be the plasma membrane. I am writing it in short forms. Students, when you are writing it, be clear what is all these representing. This is a cell wall. The next layer is going to be the plasma membrane and 2 cells. So this is going to be the cell wall. This is the plasma membrane. This is actually the next cell that is connected to this cell. The connection is through a structure called middle lamella. Hope you remember how middle lamella is formed. I have explained it to you in cell cycle and cell division unit. And coming to the inner part, this is the cytoplasmic part. So this is the cytoplasm of a plant cell. This is the single large vacuole, and substances inside the vacuole will be called as sap, and the membrane of the vacuole is called as tonoplast. And these structures what I have drawn here are the cytoskeletal structures. Cytoskeletal structures, the details we'll see it later. So these are cytoskeletal structures, and this one is the nucleus that is present in the periphery of the cell. It is present in periphery because this large vacuole pushes the nucleus to the periphery, and this lines that is randomly drawn here represents the chromosome or the chromatin fibers, and this one is the nucleolus, and the nuclear membrane extends into structures called endoplasmic reticulum. If it has ribosomes on it, these endoplasmic reticulum will be called as rough endoplasmic reticulum, and if they don't have it, it is called as the smooth endoplasmic reticulum. And this is Golgi body. Golgi body of plant cells is also called as dictyosomes. So dictyosomes is the other name of the Golgi body of plant cells alone, not for animal cells. And this structure what we have drawn here is mitochondria, and these are the plastids. The most common plastid present is the chloroplast, and these structures, the small circular structures what we have represented here, represents to microbodies. We will see in detail what are microbodies, what are different types of microbodies also. And coming to the external surface again, I have given a gap here. So this is one cell, this is another cell. There is a gap that is formed between these two cells. This gap that is seen, which can be called as connections between this cell and the next cell, this one and the other one, this is called as a pit. And this pit formation we will see when we study about cell wall. And this darkened structure what I have drawn here, this is called as the Plasmodesmata. So it is actually a cytoplasmic connection between one cell and the other cell that passes through a pit. So I have to show a difference between what is a pit and plasmodesmata, I have drawn it here. So this is a pit which is formed because of differences in their cell wall, and through a pit these cytoplasmic strands are formed to connect between one cell and the other cell. So these are the basic characteristic of a plant cell and their organelles present in them. We will study about all these organelles and their characteristics in the later classes. Coming to the structure of an animal cell can be drawn in any shape because they do not have a cell wall, and their outermost cover is going to be the plasma membrane. And the inner part we call it as the cytoplasm. And the organelles when we consider, this is going to be the centrally placed nucleus with the chromosomes or the chromatin fibers and the nucleolus. So nucleolus is clearly seen in case of plant cells and animal cells. And this structure that is always present closer to the nuclear membrane is the Golgi body or the Golgi complex. And these structures, these are lysosomes, and simply I have put small circles which represents the vacuoles. So vacuoles, they are simpler structures, they are small in number, they are present many in number also. And the continuation of nuclear membrane as endoplasmic reticulum, which can be rough or smooth endoplasmic reticulum. And these structures are the cytoskeletal structures, which give strength to the cell. And here I have given you a bigger structure, but actually in the cell they look very small. This is a centrosome with two centrioles inside. So the outer cover, the whole structure, outer cover with the centrioles, you call it a centrosome, and these two structures that are present inside, they are called as the centrioles. So these are the basic characteristics or the basic organelles of the eukaryotic animal cell, and we'll in detail we'll see about all these structures. So now we'll start with the organelles. The first part that we are going to study now is the cell wall of the eukaryotic cell, that is more specifically the plant cells. In eukaryotes, the cell wall is seen in all plant cells. It is seen in fungi. So fungi is also an eukaryotic organism, and few protists, not all protists, because there are few animal-like protists which do not have their cell wall. So plant cells, fungi and protists, few protists have their cell wall. Characteristics of their cell wall is going to be the fungi. In fungi, the cell wall is made up of mainly chitin, whereas in case of plants and protists, the cell wall is mainly made up of cellulose and hemicellulose. These are the major substances that are present in the plant cells, and there can be other substances in the plant cells like depositions of substances like pectin, lignin, suberin, cutin, etc. These are depositions that can be seen, but the cell wall is going to be made of cellulose or hemicellulose. These substances usually form depositions. Apart from these sugars, plant cells also contain lipids, few proteins, and water. So these are substances that are making up a plant cell wall. Usually the plant cell wall is made of a hemicellulose matrix, and this matrix have cellulose macrofibrils that are deposited in them. So cellulose macrofibrils forms deposits in the hemicellulose matrix. So what is this cellulose macrofibrils? Actually, macrofibrils are formed by cellulose molecules in a different way. Nearly 6,000 glucose molecules join together to form a single cellulose submolecule, and this many cellulose molecules join together to form structures called micelles, and many micelles join together to form structures called microfibrils, and many microfibrils get packed into one another to form structures called macrofibrils. So these are macrofibrils actually made of many simple cellulose units, and this macrofibrils gets deposited in the hemicellulose matrix. And the structure that connects two cell walls of two cells you call it as middle lamella, and middle lamella mainly is made of calcium pectate. The cell wall that is formed can be called as, so we can also call it as types of cell wall. This is based on their depositions. The cell wall is usually called as primary, secondary, and tertiary cell walls. Based on their differences in their depositions, they are called as primary, secondary, and tertiary cell walls. First one, the primary cell wall. Primary cell wall is going to be present in all plant cells. When I write it all cells, it is not that defining all the cells. It is going to be present in all plant cells, and they have more hemicellulose and less cellulose in them, and they are going to be a simple layer. And the secondary cell wall. Secondary cell wall is formed only in cells that require support, or that function in cells that functions as supporting cells. So only in those cells secondary cell wall would be formed, and the secondary cell wall depositions have less hemicellulose and more cellulose. This makes it more stronger than the primary cell wall. And this secondary cell wall, when it is formed, it is deposited as 3 layers. Most commonly 3 layers are formed, which are called as S1, S2 and S3 layers. Diagrammatically I will show you how this S1, S2 and S3 are formed. And secondary cell wall can have depositions. Most common depositions that are seen are containing pectin, lignin and cutin will be absent, suberin depositions can be seen in secondary cell wall. And secondary cell wall formation helps in formation of pits also. Pits can be simple pits or bordered pits. I will explain you with the diagram what is simple and bordered pits. Coming to the next type of cell wall, that is tertiary cell wall. Tertiary cell wall is usually formed in cells again that require extramechanical support, and the cells in which the tertiary cell wall is formed usually have lignin and xylem depositions, and these depositions make the cell sclerenchymatous, that is, they kill the cell, making it a dead cell because of their deposition. And here the deposition is going to be random deposition, or they are going to be spirally deposited, finally making the cell into a sclerenchymatous cell. So sclerenchymatous cells means the cells are going to become dead because they lose their cytoplasmic part. Main functions of a cell wall. Functions of cell wall, when we consider, the functions of cell wall is going to be support, giving support to the cell and in turn rigidity to the plants. It is going to be support to the cell and rigidity to the plants so that the plants grow upright. And one more main function is going to be, they are going to help in the cell wall directly involves in transport of water. This one we will see it when we study about transport in plants. Now we will see how the cell wall deposition actually happens. So when we say this is, if this is a cell, and we have seen plant cells when they are divided into two, the first formed structure is going to be a middle lamella. So middle lamella is going to be formed first. So this is going to be the middle lamella that is formed first, and on middle lamella the primary cell wall gets deposited. Once primary cell wall gets deposited, this is the primary cell wall, and once primary cell wall is deposited, these two cells are going to act as individual cells. So we will consider only this part of the cell. So this part which we are putting it in block is the middle lamella between two cells, and immediately next layer is going to be the primary cell wall of those two cells. So these two are the primary cell wall. So primary cell wall deposition is going to be first hemicellulose matrix deposits on this middle lamella, and then the cellulose macrofibrils gets inserted into that matrix, forming the primary cell wall. Next, the cell wall, the second cell wall that is formed is going to be secondary cell wall, and we know secondary cell wall is formed in S1, S2 and S3 layers. The first deposited layer is S1. The first deposited layer is going to be like this, that is, it gets deposited on the primary cell wall. So this part is going to be the primary cell wall. This one is the S1 layer. The next deposited is going to be the S2 layer, and the last deposited is going to be the S3 layer. So this is how they get deposited. So initially first S1 layer is formed, and then is going to be the S2 layer, and then is going to be the S3 layer. So they are formed one after the other. It is not that all the three layers get deposited at a time. And why I have given this spaces, so we have seen this secondary cell wall is going to be irregular in deposition, that is, they are not complete, they are incomplete layers where only primary cell wall is seen and secondary cell wall does not get deposited in this place. The place where secondary cell wall deposition is absent, this part you call it as a pit, and the pit is usually common for two cells, adjacent cells. Wherever this cell is forming a pit, in the same place its neighboring cell is also going to form a pit. So this is pit, and through this pit there can be thickened cytoplasmic part that is traveling through this pit, and this thickened cytoplasmic part is called, so I am just drawing it, I am writing it here. So this part is pit, and the thickened cytoplasmic part that moves through the pit, you call it as Plasmodesmata, which forms connection between two cells. So this is S1, S2, S3 layers which together make up the tertiary cell wall. So this is the primary cell wall, this part is the primary cell wall, and this part is going to be the secondary cell wall, and the gap where there is no secondary cell wall deposition, you call it as pit, and cytoplasmic connections, you call it as Plasmodesmata. If suppose, so this is the middle lamella layer, the primary cell wall, and the secondary cell wall deposits like this. That is, if the diameter of the pit is even from here to here, if this diameter is, so this diameter, if it is the same from S1 and S2 and S3 layers, the diameter is same, you call it as a simple pit. And when we see here, the diameter is going to be different. Diameter of S1 is less, diameter of S2 is little more, diameter of S3 is little more increased. Such type of pits, you call it as bordered pits. So this is the difference. The diameter of the pit makes the difference between the type of pit, simple and bordered pits, and cells can form any type of pits. Both simple or bordered type of pits can be formed. The next part is the plasma membrane. Plasma membrane is also called as the biomembrane, or commonly called as the cell membrane. And the main properties of plasma membrane are, they are going to be made of proteins and phospholipids, and they are selectively permeable membranes. So these are the two main properties of a plasma membrane. Based on the proteins and phospholipids that is making up the plasma membrane. So proteins and phospholipids make up the plasma membrane. So based on the arrangement of proteins and phospholipids, different models of plasma membranes were proposed by different scientists. So the models proposed were the first one, the first model, it is called as sandwich model. Sandwich model was proposed by scientists called as Danieli and Davson, and one more model called as unit membrane model. This model was proposed by scientist called Robertson. And the third model, that is the most accepted model, called as the fluid mosaic model, was proposed by scientists Singer and Nicholson. So these two scientists proposed a fluid mosaic model, and this is the most accepted model. And these two models, they were not able to explain the selectively permeable characteristic of the plasma membrane. So these two were actually rejected. The models were actually rejected, and the most accepted model of the plasma membrane is the fluid mosaic model. And according to fluid mosaic model, that is according to the concept given by Singer and Nicholson, the proteins that are present in the plasma membrane, they are going to be of two types. They are intrinsic proteins and extrinsic proteins. The intrinsic proteins are those that are completely embedded in the lipid layer. Such proteins are called as intrinsic proteins, and extrinsic proteins, they are found on the surface of the lipid layer. Such proteins are called as extrinsic proteins. And the phospholipids that are present are going to be present in bilayer. So the two layers are actually held together by weaker forces like van der Waals forces, and the bilayers are going to be amphipathic in nature. Amphipathic அப்படினா amphipathic characteristic அப்படினா என்னன்னா ஒரு layer, it is going to have two ends, and the two ends are going to be opposite in their characteristic. இப்போ amphipathic character அப்படினா என்ன அப்படினா இந்த lipid வந்துட்டு it has, அவங்க சொன்னது என்னன்னா it has a head and a tail. Head is going to be hydrophilic in nature, and the tail is going to be hydrophobic in nature. So இதுதான் வந்துட்டு ரெண்டு characteristics அவங்க சொன்னது. So they are going to have a head and tail, that is two ends. So amphipathic அப்படினா ரெண்டு இருக்கணும், ஒன்று வந்துட்டு ends, ரெண்டு ends இருக்கணும், and these two ends are going to have different opposite characteristics. Hydrophobic அப்படினா water hating characteristics, that is they move away from water. Hydrophilic வந்துட்டு water loving, means they are going to interact with water molecules. So it is usually represented like this. So this is going to be the head, which is hydrophilic in nature. This is going to be the tail, which is hydrophobic in nature. To the head, a phosphate molecule would be attached, so making it a phospholipid layer. And this bilayer, it is going to be represented in the form of the lipid bilayer like this. So fluid mosaic model shows that the proteins or intrinsic and extrinsic proteins that are embedded in the lipid layers. So lipid is first going to be in bilayers. So we are representing the bilayers like this, and intrinsic proteins are those which are present within the lipid layer. So within the lipid layer, the proteins are going to be embedded. So this is an intrinsic protein. Extrinsic protein, when you consider, they are usually found on the surface of the lipid bilayer. So these are intrinsic proteins, these are extrinsic proteins, and this is going to be the lipid bilayer. And how do this lipid bilayer explain the selectively permeable characteristic? Yes, selective permeability means it allows the movement of the plasma membrane, is going to allow the movement of substances based on their need. So they are going to select the substances which are required, and they are allowing the substances to move in and out of them. And the movement of substances is because of the flexibility of the plasma membrane, and the flexibility characteristics is given by the lipids. The lipids that are present in the lipid bilayer, they are going to exist in form of quasi-fluids, and these quasi-fluids are more or less jelly-like substances, and these lipids, they allow the movement of the proteins itself, that is the intrinsic and extrinsic proteins. It is not that the intrinsic protein has to be always remaining in the same position. According to fluid mosaic model, these intrinsic proteins can move from one position to the other. Similarly, extrinsic proteins, they can move in and they can move out also. That is, they are allowing the movement of this intrinsic and extrinsic proteins. So because they are semi-fluidic in nature, they become flexible, and because of movement of proteins, they allow the movement of substances, making it selectively permeable. Thus, the fluid mosaic model given by Singer and Nicholson is going to explain both the properties, that is, they have proteins, phospholipids, and they are selectively permeable also. And coming to the functions of the plasma membrane. Plasma membrane helps in transport of substances in and out of the cell, and the transport can be active or passive, and they are also helping in attachment of the cells to the neighboring cells. So attachment is with the help of substances called as junctions. This we'll study about junctions, types of junctions in detail about when we study the unit structural organization in animals. Similarly, different types of transport like active passive transports, diffusion, facilitated diffusions, pumps, etc. we'll be studying it in transport of plants unit. So this one explains you the characteristics of a plasma membrane and the fluid mosaic model of plasma membrane. And the fluid mosaic model of plasma membrane, though we have studied it in the eukaryotic structure, it is going to be same for prokaryotic and eukaryotic cells. The next part of a cell is the cytoplasm. The cytoplasm is a jelly-like or a semi-fluidic translucent substance. So where is this cytoplasm present is its location. More specifically, we can say it as between nuclear membrane and plasma membrane. So நம்ம ஒரு செல்லை கன்சிடர் பண்ணுறோம் அப்படின்னா, if this is the cell, the cell நியூக்ளியஸ் இங்கே இருக்குது, இந்த பார்ட் இங்கே இருக்கிற சப்ஸ்டன்ஸ் மட்டும்தான் வந்துட்டு we call it as cytoplasm. இதுக்குள்ளே இருக்கிறது வந்துட்டு நியூக்ளியஸ்க்குள்ளே இருக்கிறது வந்துட்டு நியூக்ளியோபிளாசம். ஸோ சைட்டோபிளாசம் அது பிளாஸ்மா மெம்பரேனுக்கும் நியூக்ளியர் மெம்பரேனுக்கும் நடுவில் இருக்கிற பார்ட் சைட்டோபிளாசம். நியூக்ளியஸ்க்கு உள்ளே இருக்கிற பார்ட் வந்துட்டு நியூக்ளியோபிளாசம். சைட்டோபிளாசம் நியூக்ளியோபிளாசம் டுகெதர் we call it as protoplasm. ஸோ protoplasm அப்படின்னா என்னென்னா சைட்டோபிளாசம் ப்ளஸ் nucleoplasm. So this one together you call it as the protoplasm. So cytoplasm vandittu it is present between nuclear membrane and plasma membrane, and cytoplasm can be divided into two parts. One part is called as the cytoplasmic matrix. The second part is called as cytoplasmic structures. So cytoplasmic matrix is actually the semi-fluidic substance. எதில் வந்துட்டு cytoplasmic structures இருக்கோ அந்த semi-fluidic structureக்கு பேர் வந்துட்டு matrix. இப்போ இந்த semi-fluidic structure அந்த matrixோட function என்ன அப்படின்னா, it is the site of all metabolic activities. இன்னொன்று they help in formation of pseudopodia. எப்படி pseudopodia ஃபார்ம் பண்ணுறதுக்கு ஹெல்ப் பண்ணும் அப்படின்னா, சப்போஸ் வில் கன்சிடர் இந்த பிளாஸ்மா சைட்டோபிளாஸ்மிக் மேட்ரிக்ஸ் இருக்கு இல்லையா, இந்த மேட்ரிக்ஸ் வந்துட்டு மேட்ரிக்ஸ் தட் இஸ் க்ளோசர் டு பிளாஸ்மா மெம்பரேன். அதாவது இந்த இடத்துல இருக்கிற மேட்ரிக்ஸ் பார்ட் சைட்டோபிளாஸ்மிக் கண்டென்ட் ஆஃப் that is present closer to plasma membrane. இந்த பார்ட்டில் இருக்கிறது வந்துட்டு we call it as the gel or ectoplasm. Similarly, matrix ஓட inner part வந்துட்டு we call it as sol or endoplasm. இந்த ஜெல்லும் சாலும் வந்துட்டு they are interconvertible. இந்த இன்டர்கன்வெர்ட்டபிள் நேச்சர்னால தான் வந்துட்டு சூடோபோடியா ஃபார்மேஷன் நடக்குது. த இன்டர்கன்வெர்ட்டபிள் நேச்சர் ஆஃப் ஜெல் அண்ட் சால், இதனால் வந்துட்டு சூடோபோடியா ஃபார்மேஷன். இந்த சூடோபோடியா ஃபார்மேஷன் எதுக்கு வந்துட்டு செல்லுக்கு ஹெல்ப் பண்ணும் அப்படின்னா, ஃபார் எண்டோசைட்டோசிஸ் ப்ராசஸ். ஸோ ஃபுட்டை உள்ளே எடுத்துக்கணும் அப்படின்னா செல் வந்துட்டு எண்டோசைட்டோசிஸ் பண்ணும். எண்டோசைட்டோசிஸில் சூடோபோடியா ஃபார்ம் பண்ணி they are going to engulf the food. So this is the part of the cytoplasm which is called as matrix. இன்னொன்று வந்துட்டு cytoplasmic structures. Cytoplasmic structuresல again there are two parts. One is organelles cytoplasmic organelles. இன்னொன்று வந்துட்டு they are called as cytoplasmic inclusions. இப்போ cytoplasmic organelles அப்படின்னா we know they are including all membrane bound organelles. Whereas inclusions அப்படின்னா எடுத்துட்டோன்னா inclusions அப்படின்னா என்னென்னா இது வந்துட்டு non-living substances present in the cytoplasm. So non-living substances that are present, அதாவது இந்த inclusions இருக்கு இல்லையா, they are also called as ergastic substances or deutoplasmic substances. So என்னென்ன non-living substances இருக்க முடியும்? ஃபஸ்ட்டு வந்துட்டு ஃபுட். ஃபார் எக்ஸாம்பிள் பிளான்ட் செல்ஸாக இருந்தால் ஸ்டார்ச்சை ஸ்டோர் பண்ண போகுது, அனிமல் செல்ஸாக இருந்தால் கிளைகோஜனை ஸ்டோர் பண்ண போகுது. ஸோ தீஸ் ஆர் எர்காஸ்டிக் சப்ஸ்டன்சஸ். செகண்ட், மினரல்ஸ். ஸோ மினரல் நியூட்ரியன்ஸ். ஸோ இன்ஆர்கானிக் சப்ஸ்டன்சஸை வந்துட்டு ஸ்டோர் பண்ணி வச்சுருக்கிறதும் வந்துட்டு எர்காஸ்டிக் சப்ஸ்டன்சஸ் தான். தேர்ட், கேஸஸ். ஸோ செல்ஸில் வந்துட்டு யூஸ்வலாக ஆக்சிஜன் யூஸ் பண்ணும், கார்பன் டை ஆக்சைடை ரிலீஸ் பண்ணும், அண்ட் தேர் கேன் பி நைட்ரஜன் ஆல்சோ. இதெல்லாம் வந்துட்டு பேசிக் டைப் ஆஃப் கேஸஸ் தட் கேன் பி ப்ரெசன்ட். ஃபோர்த் டைப் ஆஃப் எர்காஸ்டிக் சப்ஸ்டன்சஸ் வந்துட்டு எக்ஸ்க்ரீட்ரி சப்ஸ்டன்சஸ். எக்ஸ்க்ரீட்ரி சப்ஸ்டன்சஸ் அனிமல்ஸில் எடுத்துட்டால் அமோனியா இருக்க முடியும், யூரியா, யூரிக் ஆசிட், ஹிப்யூரிக் ஆசிட். இதெல்லாம் வந்துட்டு எக்ஸாம்பிள்ஸ் ஆஃப் எக்ஸ்க்ரீட்ரி சப்ஸ்டன்சஸ் இன் அனிமல் செல்ஸ். பிளான்ட் செல்ஸில் எடுத்துட்டிங்கன்னா ரெசின்ஸ், கம்ஸ், ஆயில்ஸ், இதெல்லாம் வந்துட்டு எக்ஸ்க்ரீட்ரி சப்ஸ்டன்சஸ் இன் பிளான்ட் செல்ஸ். ஸோ இதெல்லாம் வந்துட்டு டுகெதர், ஆல் தி சப்ஸ்டன்சஸ் டுகெதர் ப்ரெசன்ட் இன் சைட்டோபிளாசம். அதை நம்ம செல் இன்க்ளூஷன்ஸ் ஆர் சைட்டோபிளாஸ்மிக் இன்க்ளூஷன்ஸ் ஆர் எர்காஸ்டிக் சப்ஸ்டன்சஸ் ஆர் டியூட்டோபிளாஸ்மிக் சப்ஸ்டன்சஸ்னு பார்ப்போம். ஸோ வித் திஸ் நம்ம we have completed the cell wall, plasma membrane and the cytoplasmic part. அடுத்த வீடியோவில் வந்துட்டு we'll see about all the organelles. So students, நீங்கள் இது வரைக்கும் வந்துட்டு we have seen about the basic structure of cell, types of cells, prokaryotic cells ஓட characteristic, eukaryotic cells ஓட comparison and characteristics, plus 3 organelles பார்த்துருக்கோம், 3 structures பார்த்துருக்குறோம், cell wall, plasma membrane and cytoplasm. அடுத்த வீடியோவில் வந்துட்டு we'll see about organelles. Any doubts you can please contact. Thank you.